There are various factors that affect enzyme activity, but before we discuss that, let us define enzyme activity. Enzyme activity refers to the measure of a rate at which an enzyme converts substrate to products in a biochemical reaction. The four major factors that affect enzyme activity are the following. We have temperature, pH, concentration of both the substrate and the enzyme. Let us discuss the first factor, temperature. This is a measure of the kinetic energy of molecules. The higher temperature means molecules are moving faster and colliding more frequently. Enzyme activity has a directly proportional relationship with the temperature. As the temperature of an enzymatically catalyzed reaction increases, the rate or velocity of the reaction also increases. But when the temperature increases beyond a certain point, the increased energy begins to cause disruptions in the tertiary structure of an enzyme, causing denaturation. The change in the tertiary structure slows down the catalytic action, thus also decreasing the enzyme activity. With that, an optimum temperature must be met. Optimum temperature refers to the temperature at which an enzyme exhibits maximum activity. The next factor is pH. Small changes in pH can result to enzyme denaturation and subsequent loss of catalytic action. Most enzymes exhibit maximum activity over a very narrow pH range. So just like the temperature, optimum pH must also be met. Each enzyme has a characteristic optimum pH, which usually falls within the physiologic pH range of 7.0 to 7.5. Just like in the nomenclature system, digestive enzymes are also exceptions in this condition. Pepsin, the active digestive enzyme in the stomach, functions best at a pH of 2.0. On the other hand, the enzyme trypsin, which operates in the small intestine, functions best at the pH 8.0. The next one is substrate concentration. When the concentration of an enzyme is kept constant and the concentration of substrate is increased, the enzyme activity pattern shown in figure 21.8 is obtained. This activity pattern is called saturation curve. Enzyme activity increases up to a certain substrate concentration and therefore remains constant. So what do we mean by that? If you could still remember in the first video presentations, enzymes are not being consumed in an enzymatic activity. When the amount of substrate increases, saturation may be met, thus decreasing the enzyme activity. As substrate concentration increases, the point is eventually reached where enzyme capabilities are used to their maximum extent. The rate remains constant from this point on. Each substrate must occupy an enzyme active site for a finite amount of time, and the products must leave the site before the cycle can be repeated. When each enzyme molecule is working at full capacity, the incoming substrate molecules must wait for their turn for an empty active site. At this point, the enzyme is said to have under saturation conditions. The rate at which an enzyme accepts substrate molecules and releases product molecules at substrate saturation is given by its turnover number. So what do we mean by turnover number? An enzyme's turnover number is the number of substrate molecules transferred per minute by one molecule of each enzyme under optimum conditions of temperature, pH, and saturation. The last factor is the enzyme concentration. If the amount of substrate present is kept constant and the enzyme concentration is increased, the reaction rate increases because more substrate molecules can be accommodated in a given amount of time. The greater number of enzyme concentration, the greater the reaction rate. So, the enzyme concentration is directly proportional to the enzyme activity. Now, let us discuss regulation of enzyme activity. This is a necessity for many reasons. Many mechanisms exist by which enzymes within a cell can be turned on and turned off. We consider, in general terms, three such mechanisms: First one is feedback control associated with allosteric enzymes. The second is proteolytic enzymes and zymogens, and the last is the covalent modification. Before we discuss the first mechanism, let us define allosteric enzymes. Take note that many, but not all of the enzymes responsible for regulating cellular processes are allosteric enzymes. All allosteric enzymes have quaternary structure, that is, they are composed of two or more protein chains. All allosteric enzymes have two kinds of binding sites, for the substrate and for the regulators. Active and regulatory binding sites are distinct from each other in both location and shape. Often, the regulatory site is one of the protein chain and the active site is on another. Then lastly, binding of a molecule at a regulatory site causes changes in the overall three-dimensional structure of the enzyme, including structural changes at the active site. Substances that bind at regulatory sites of allosteric enzymes are called regulators. We have two types, the positive and the negative regulator. The binding of a positive regulator increases enzyme activity. The shape of the active site is changed such that it can more readily accept substrate, while the binding of a negative regulator decreases its activity. Changes to the active site are such that substrate is less readily accepted. Let us see the illustration of a positive and negative regulation. As you can see, when a negative regulator attaches to the active site, the shape is being changed. Thus, a substrate now cannot enter the active site. While, for the positive regulator, the shape is changed so that a substrate can enter the active site. Let us now discuss feedback control. This is a process in which activation or inhibition of the first reaction in a reaction sequence is controlled by a product of reaction sequence. Consider a biochemical process within a cell that occurs in several steps. Each step is catalyzed by a different enzyme. The product of each step is the substrate for the next enzyme. So for this illustration, what will happen in this reaction series if the final product is a negative regulator for the first enzyme? At low concentrations of the final product, or the D, the reaction sequence proceeds rapidly. At higher concentrations of D, the activity of enzyme 1 becomes inhibited through the process of feedback, and eventually the activity stops. At the stopping point, there is sufficient D present in the cell to meet its needs. Later, when the concentration of D decreases through the use of other cell reactions, the activity of enzyme 1 increases and more D is being produced. The next mechanism is proteolytic enzymes and zymogens. A second mechanism for regulating cellular enzyme activity is based on the production of enzymes in an inactive form. These inactive enzyme precursors are then turned on at the appropriate time. Such a mechanism for control is often encountered in the production of proteolytic enzyme. This proteolytic enzyme is an enzyme that catalyzes the breaking of peptide bonds that maintain the primary structure of protein. Proteolytic enzymes are generated in an inactive form because they would otherwise destroy the tissues that produce them. Later on, when they are needed by our body, they will be converted to their active form. They are called zymogens. Zymogens are the inactive form of the proteolytic enzyme. Activation of a zymogen requires enzyme-controlled reaction that removes some part of zymogen structure. Such modification changes the three-dimensional structure of the zymogen, which affects the active site conformation. Let us now discuss covalent modification of enzyme. Covalent modification is a process in which enzyme activity is altered by covalently modifying the structure of the enzyme through attachment of chemical group to or removal of a chemical group from a particular amino acid within the enzyme structure. The most commonly encountered type of covalent modification involves the processes by which a phosphate group is added to or removed from the enzyme. This phosphorylation or dephosphorylation process is the off-on or on-off switch for the enzyme. Phosphorylation is the process of addition of the phosphate group to the enzyme by protein kinases, while dephosphorylation is the removal of the phosphate group from the enzyme by phosphatases. Let us now discuss antibiotics that inhibit enzyme activity. An antibiotic is a substance that kills bacteria or inhibit their growth. Antibiotics exert their action selectively on bacteria and do not affect the normal metabolism of the host organism. Antibiotics usually inhibit specific enzymes essential to the life processes of bacteria. The first one is sulfadrugs. Sulfanilamide inhibits bacterial growth because it is structurally similar to PABA or para-aminobenzoic acid. Many bacteria need PABA in order to produce an important coenzyme, which is folic acid. Sulfanilamide acts as a competitive inhibitor to enzyme in biosynthetic pathway for converting PABA into folic acid in this bacteria. Folic acid deficiency retards growth of the bacteria and can eventually kill them. The next one are penicillins. All penicillins have structures containing a 4-membered B-lactam ring fused with a 5-membered thiazolidine ring. Penicillins inhibit the enzyme transpeptidase, an enzyme that catalyzes the formation of peptide cross-links between polysaccharide strands in bacterial cell walls. This cross-link strengthens cell walls. By inhibiting the enzyme transpeptidase, penicillin prevents the formation of a very strong cell wall in the bacteria. Any osmotic or mechanical shock then causes lysis, killing the bacterium. Penicillin is highly specific in binding to the active site of transpeptidase. In this sense, it acts as a very selective competitive inhibitor. However, unlike a normal competitive inhibitor, once bound to the active site, the beta-lactam ring opens as the highly reactive amide bond forms a covalent bond to a critical serine residue required for normal catalytic action. The result is an irreversible inhibited transpeptidase enzyme. The next drug is ciprofloxacin. The antibiotic ciprofloxacin hydrochloride, or marketed as Cipro, is an effective agent against bacterial infections in many different parts of the body. It is effective against skin and bone infections, as well as against infections involving the urinary, gastrointestinal, and respiratory systems. It is the drug of choice for traveler's diarrhea. It is considered one of the best broad-spectrum antibiotics available. Bacteria are slow to acquire resistance to ciprofloxacin, unlike penicillin. So what are the different medical use of enzymes? Enzymes can be used to diagnose certain diseases. Although blood serum contains many enzymes, some enzymes are not normally found in the blood but are produced only inside cells of certain organs and tissues. The appearance of these enzymes in the blood often indicates that there is tissue damage in an organ and that cellular contents are spilling out into the bloodstream. That concludes our discussion on enzymes.